start your talk. Yeah.
Okay, can you hear me?
Yeah.
Okay, excellent. So thanks for the Ru-Ching for organizing this awesome virtual symposium. I'm honored to be here. I also wish Kurt Simic a quick and full recovery and look forward to seeing his presentation in 2 weeks. Happy that I could fill in this slot. This morning, at least in my time zone, I'll present a bit of EM history from the past to the present and summarize the 6-year history and where I think I see some of the future of fully programmable EM preparation of specimens and grids is going. So some history to put this into context. Biological EM began due to these and many other EM pioneers. So we're beginning at the beginning here. They created the chemical preparation workflow that we use today, aldehyde fix, osmium post fix, something that I think all of us here know quite well. Let me get the laser pointer up. Okay. So this is where it all began. We can take another look at this workflow here, and basically the workflow is a sequence of reagent exchanges. And this is where in sample preparation, we spend most of our active time in the laboratory as we're moving from an initial fix through rinsing, label, staining, post-fixing, et cetera, and then another series of reagent exchanges for grid prep, especially for processes like immunogold. This can take a lot of time and effort. So we can take a look at this here, and this is the way these reagent exchanges were done back at the beginning of EM prep. They used glass vials and pasteur pipettes to do the reagent exchanges. If we fast forward to today, and beginning in the 1970s, the devices that are used for these reagent exchanges in most laboratories are pretty much the same. These have changed very, very little. And I believe this would also include the EM autoprocessor and the microwave oven, which was first introduced in around 1970 by Myers and later refined by Giberson and others through the 80s, 90s, and 2000s. But the most fundamental process, reagent delivery, really has changed very little over the last 50 years or so, with this 50 years of this history. So with this history, let's look into why we created a new system for automated and manual EM sample preparation. Well, we wanted to streamline the reagent exchange workflow. This is where we spend most of our time in the laboratory. We would love to have more reproducible results, consistent results. Of course, we would love to ease our workflow. And ideally, it would be nice to be able to prepare specimens and grids while we're at the scope, at the microtome, or dealing with those myriad office tasks that we always have to deal with. We also want to enable really highly complex and time-consuming protocols. Immunogold labeling, IGL, is certainly one of those. And a more recent technique that's becoming increasingly important are the three-dimensional electron microscopy methods like serial block face SEM, FibSem, and array tomography SEM. These require an extensive amount of time. Well, how complex are these and how time consuming? Where are we going to reap benefit from being able to streamline and even automate this workflow? So again, we can take a look at this, this sequence here. And we have a lot of time and effort in preparing specimens. And in many experiments, principally immunogold labeling, we even have more specimens to track where we might even have different reagent flows. We have controls, we have comparisons, we have titrations. So basically, the short answer is these complexities and time can be very, very extensive. So we can take a look at some of these. And of course, these are just single snapshots of the different methods. Every lab is different. But for a typical TEM of a tissue, we're talking about 19 reagent steps from fixatives through all of the rinses and all the other reagents. In most labs, this is something like a 1 to 3 day process. The hands-on effort that we're spending at the fume hood is about 8 hours, or about 3 hours of very busy exchanges if we're doing microwave processing. And sometimes we even have more specimens to deal with, like controls and such, that we need to treat a little differently. When we go to immunogold labeling for on-grid, this is about 27 steps. Pretty typically takes most labs about a day to perform this, about 8 hours of actual effort, and we typically are tracking at least two conditions, and maybe even 3 or 4 or even more conditions. If we're doing pre-embedding immunogold labeling with silver enhancement, we're talking about 46 different reagent steps. That's a lot. And typically, after we do the immunogold labeling, we're going to be then embedding the sample for TEM. So we're talking about a 2 to 4 day process. And if we add up the hours, we're looking at about 27 hours of hands-on time. 3D-EM, even more steps than immunogold, even more time. And if we want to do pre-embedding silver enhancement immunogold before we are preparing the sample for 3D-EM, we're looking at 46 steps plus 63 steps over about 2 weeks of active time, or about 32 hours of very busy technical time. So this would probably explain why, at least in my searching, I have found extremely few reports of silver enhanced immunogold labeling followed by 3D-EM like serial block phase or FibSem. I think I only found one very clear paper in this side of things. So this is why we try to streamline this process here. So that's really where we have the foundation of what we call the MPrep system. So this was introduced just 10 years ago. And the core MPrep system consists of capsules for specimens and grids that are used for both manual preparation and automated preparation. And for those not familiar with them, they're quite simple. A specimen is simply placed into an MPrep as micropreparation specimen capsule, entrapped with a screen, which can then be removed and adjusted as necessary, or grids are attached or are inserted into an MPrep G capsule, a grid capsule, and they're entrapped there by a simple pinch function that requires a little bit of practice, but is quite easy to do once you've done this. So once the grids or the specimens are inserted into the capsules, in many applications, you don't even have to touch them again. You can do all the processing. How does the processing occur? The capsules are just attached to an ordinary pipette man for either the specimen capsules or the grid capsules. So the capsules function like pipette tips, but uniquely the specimen is entrapped inside the tip, sort of reversing the way it's done in a standard biotech lab with the tips. So by using these pipetters, we can utilize microwell plates and other apparatus that are used for creating streamlined workflows in the biotech laboratory. And we can choose different types of microwell plates to draw the reagents, to pipette the reagents up from, and that gives us a lot of flexibility. So reagent exchanges, even manually, are very easy. Simply depress the button one time, and you've now exchanged the reagents on 16 grids, 2 grids per capsule, or 8 specimens. Very, very fast and easy and uniform. So that's useful, but it's not automation. So at M&M 2015, just 66 years ago, we introduced the MPrep ASP 1000 automated specimen processor. We created the ASP to meet the needs of small and large labs for specimen and grid preparation to provide ease and speed for routine tasks, including the reproducibility of fully automated preparation. We tried to create a system with logical protocol programming. We wanted to create a system that had more than enough reagent steps for the most complicated protocols, such as immunogold labeling and serial block phase SEM and other 3D-EM preparation. We wanted to have low reagent consumption when that's important and necessary, say for applications such as immunogold labeling with very expensive antibodies. So we've achieved this with just 35 or so microliters per grid, for example. And we wanted to have the right number of specimens and grids for most applications in most labs. So the ASP 1000 looks and works like this. I also want to say we can also implement fast preparation. So the ASP looks like this. So this is the pipetting head, and the specimens, here specimen capsules are loaded onto the pipetting head. And there are 8 capsules for 8 specimens shown here. Grids mount the same way. I'll show that later. The reagents are loaded into microwell plates. Here, this one is labeled for the different reagents. And each capsule follows a different path through these well plates. So this first uh, capsule will draw in reagent processing reagents from the first row in the microwell plate. And in that way, each specimen can, when you desire it, receive a different sequence of reagents. So you can accommodate those controls and things. So there are positions for 6 plates and trays. So one can do a lot of different reagents along this way. The plates can be covered or sealed to reduce evaporation, as you can see in this photo in here. You can use different types of plates to vary the volume. Obviously, lower volume plates for expensive reagents and more for rinses. And you can organize the plates in any way you desire for a particular preparation protocol. For example, many labs place all the heavy metal reagents in one plate in order to simplify the waste stream handling. And you can even change the plates or add a reagent during a protocol. The ASP can text you when this step is needed, and this enables even more than 72 steps, such as I pointed out earlier, doing pre-embedding immunogold labeling followed by serial block-phase TEM preparation. The protocol program specifies the reagent by its location. Where is it in a plate? It specifies the time, the agitation, and with an ASP accessory, the temperature of the reagent tray. In the 6 years since the ASP 1000 introduction, many protocols have been developed by ASP users and by us working with many users, and I'll survey these in the next several slides. These 3D EM and TEM preparation protocols also can provide extremely fast tissue preparation by using very gentle but continuous reagent mixing to accelerate the diffusion of processing reagents into the tissues. And of course, also implementing automated immunogold preparation. This is typically done more slowly. So let's first take a look at TEM specimen preparation as I survey these things. So most applications, not surprisingly, have been for mammalian tissues. Here are TEM images of rat kidney and rat liver. Both specimens were prepared using the same very rapid 2¼ hour protocol. And this is the time from after overnight fixation in a Karnofsky's formulation until the resin infiltrated specimen still in the MPrep-S capsule was transferred to the oven for resin curing. The ASP protocol program and many others, this particular one and many others, were developed by Microscopy Innovations, working with the School of Medicine and Public Health at the University of Wisconsin in Madison, as you'll see here. So the first published demonstrations of rapid tissue preparation with the ASP was by Melania McLean at the Stowers Institute for Medical Research. She wanted to accelerate the slow 7-day manual protocol they used to prepare planarium flatworms. In this study, she sped this week-long protocol by employing extremely fast ASP agitation to create an 8-hour protocol, which actually included fast epoxy curing, and as shown here, achieved comparable results with the 7-day protocol. Another application of a non-mammalian specimen is shown here of yeast, which was agar enrobed following glutaraldehyde fixation and pelleting, then placed into the capsules and subsequently processed using the ASP in 1½ hours before resin curing. Skeletal muscle it has also been prepared. This particular study by Tetri et al. Was published in Frontiers in Physiology. And we can see they were actually examining altered mitochondrial structure in this particular study. I'll talk a little bit more about that at the end. And here we see cardiac muscle in the upper panel and a brain synapse with lots of synaptic vesicles here, and that was prepared in 1.5 hours. Here are 3 examples of tissues prepared using LR white acrylic embedding without osmium post-fixation, as you would want to do for on-grid immunogold labeling. So we have liver, mouse neuronal somas, dorsal root ganglion, and mouse extracellular muscle. Now let's take a look at immunogold labeling, both on-grid and en bloc or pre-embedding. So this study, which was the first ASP automated immunogold labeling work, used C. elegans specimens that were first high pressure frozen, freeze substituted, and then embedded in an LR white acrylic uh, embedment prior to sectioning. The sections were then placed on grids, inserted into grid capsules, which were then placed here, placed in the on the ASP head. Reagents were arrayed here on the tray and processed using Orion reagents and the Orion protocol in 7.5 hours. So there was no attempt to speed up the labeling study in this particular study. This came actually from the laboratory of Ru-Ching. And in this work, we see pre-embedding mouse brain with silver enhancement. And this publication described the process as a 60-minute setup, a 19-hour runtime. There was a pause in the program for the operator to add the silver enhancement reagent, which can be quite labile, and then was continuing process. On grid was also demonstrated in the first of these works here. And here we have sections of chlamydia infected HeLa cells that were labeled and whole mount preparation of a bacteria with an anti-flagellar antibody. And again, this paper described a 30 to 60-minute setup, 6 3/4 hours runtime, which is often done overnight. I want to point out that they spent a lot of time on setup and then basically walked away. We can also now look at serial block phase SEM, and these are the last set of examples I'm going to show you. So Benson and colleagues at the Cleveland Clinic, initially working with us and now certainly often running with these methods, were the first to demonstrate serial block phase 3D EM prep of brain with the ASP. Here they compared their 5-day manual protocol to a rapid 1-day protocol using samples from the same brain, the same Karnofsky's fixed brain. So we have identical samples where they looked at the cortex and they observed high quality preparation, where they reported comparable metal staining, preservation, infiltration, sectioning, and imaging of myelin synaptic vesicles and mitochondrial cristae. So for this work, actually, Emily Benson received the first place Diatome Award last year at M&M. So qualitatively, the prep looks comparable as they have assessed here, but Benson and colleagues wanted to compare this more quantitatively. So for that, they did a comprehensive study of axon and myelin dimensions. And as they point out, axon and myelin dimensions are very sensitive to changes in fixation and staining. And basically, they wanted to confirm that they could use this automated approach with the same kinds of data sets that they had already done manually. So they analyzed nearly 5,000 axons measured, as we see here, color-coded in here. And what they reported, what they found, were that axon diameters were comparable, myelin thickness were comparable, not statistically significantly different, and something called the G ratio, which is the ratio of axon to myelin diameter, were all not statistically different at all between manual and ASP prepared specimens. So that convinced them that this is quite viable. So as a microscopist, I'm just thrilled with the power of three-dimensional EM. And so in this particular work, they were able to use the 3D volume data to extract the structure of synapses. So here we see a dendrite that's color-coded in one particular plane from the 3D EM data set. We see three different synapses, and they can reconstruct the three-dimensional structure of the synapses. In this one, they show the synaptic vesicles in the reconstruction, and in these two, they look at the mitochondria. And what is fascinating is that the mitochondria and the presynaptic terminal are actually intertwined around each other. And this takes a couple of different views to see this, something that's very difficult to see in a single section view. Benson and colleagues also compared rapid ASP preparation to standard preparation of liver. And again, they found the quality were comparable between the two. Way back in 2017, Melania McLean also did a comparison study of manual and ASP preparation. This study compared a 7-day manual to a 1-day automated prep for planaria and reported comparable results. For this work, McLean also received a Diatom Award, and about 2 years later used this protocol to enable the 3D reconstruction of dividing cells within a planaria. And finally, this will be the last study I'll show. This was done by the Department of Pediatrics at the University of Wisconsin, working with me at Microscopy Innovations and some others, and also with Thermo Fisher. And they use serial block phase SEM to examine the effects of neonatal incubators on cardiac right ventricle mitochondria. They did this to understand why children and adults who were in NICU incubators have reduced exercise and oxidative capacity, even again, as they are young children and moving into adulthood. So the study compared normal 3-week-old rats, normoxic, to hyperoxic rats that spent 2 weeks at the high oxygen levels of a neonate ICU incubator. These hyperoxic treated rat pups had a lowered volume percent of mitochondria, as you can see here. And I think even more striking, they had very different mitochondrial shapes, which were able to be reconstructed from the three-dimensional volume data sets, as we see partially here. And we can qualitatively easily see that these mitochondrial shapes are very different between the normal conditions and the hyperoxic treated rat pups, and we can quantitatively demonstrate this in terms of volume and shape are very, very different. Again, the tissue prep was done in just 1 day instead of a standard day manual protocol, thus accelerating the time to knowledge and achieving this with less effort. And again, I want to acknowledge here, all the imaging and image analysis was done by the Thermo Fisher Nanoport facility. So I want to summarize what I've shown today here. So we have demonstrated TEM tissue prep using kind of standard glutaraldehyde osmium epoxy preparation. We've demonstrated glutaraldehyde acrylic preparation with an acrylic resin. And we've demonstrated serial block phase 3D EM prep, and as well as immunogold labeling. And with all of these, it was faster and much less effort. Not necessarily faster with the TEM, but much, much less effort. So there are many protocols that now have been developed, and with this library of protocols and library of users, It's now time to better facilitate protocol sharing among users. And this is someplace we're going right now at the company here. So each ASP protocol, which consists of a reagent processing sequence, is a small file that's easily shared even via email. But this method doesn't reach the entire ASP user community. It only reaches those individuals who are in the email chain. So we're in the process of creating a cloud-based sharing system that will facilitate sharing among all the users that choose to do so. And of course, all users would be welcome to share these here. But there's more to an EM prep protocol than just the reagent sequence as encoded in the protocol. So to facilitate cloud sharing, we want to be able to also be able to share additional protocol data. Uh, some of it's pretty obvious. What specimens is it good for? Who developed it? Who's used it? Are there any publications associated with this work? But the most complicated part of this is the reagent info. And as I pointed out earlier, people who are doing automated preparation spend most time on doing the setup which largely consists of filling the reagent trays. So let's kind of take a look at that. So we want to be able to share these folders and particularly the reagent exchanges. So the reagent info is, as I said, more complex. So we want to put this into a format that it will be useful for MPrep system users. So we can provide, or we're beginning to provide, these downloadable templates that provide a format of how to fill the microwell plates used to hold the reagents. And this is for both a 96 well plate wells or 12 well templates. And the user simply fills these out and can color them in as they need to, and these will provide a guide. And so they can fill it out for it. In this case, an on-grid immunogold labeling protocol with titrations and controls all filled out and additional notes. And this particular note for a final dehydration and resin plate layout for final dehydration and resin infiltration, where all the specimens receive exactly the same reagent sequences. And to use these, once these are filled out, it's a simple matter of placing a plate on top of the template and using this as a color-coded guide for the filling to make this job much easier, as easy as possible. And again, you can provide whatever notes you desire here. So with this, the goal is to be able to share these into these same folders and have them uploaded to enable a library of sharing. So this is something that we are now pursuing, and we hope to further help the microscopy community with this capability. So again, we have programmable automated preparation, and since the introduction, dozens of protocols have been demonstrated, which ease workflow, enable complex and time-consuming protocols, and can facilitate easier protocol sharing with even easier sharing in process. With that, I want to acknowledge Tom Strader, who is here. Hi, Tom. Tom was the ASP 1000 developer, product manager, and VP of R&D at Microscopy Innovations. I want to acknowledge Jeff Percival, who's the ASP 1000 software engineer and developer. And of course, I must acknowledge all the customers and collaborators, especially as I've presented here. So this includes Melania McLean and Morgan Harwood at the Stowers Institute, Ru-Ching, thank you very much, Emily Benson and Graham Kidd at the Cleveland Clinic, multiple individuals at the University of Wisconsin, and multiple individuals at the Thermo Fisher Hillsborough Nanoport facility. Who were instrumental in the last study that I showed. I'd like to also acknowledge our many other collaborators and users of the system, some of whom are shown here, and this includes Kurt Simic at the Danforth Plant Sciences Center. So I'll conclude here with thank you for your attention. Do feel free to reach out to me via email, our website, and in our discussion section later this morning, at least at my time zone. And I want to call your attention to a discussion we're hosting next week. What's with automation in EM core facilities? Talk to folks who have done it. And with that, I want to acknowledge our invited guests who have graciously acknowledged or graciously agreed to be available for a Zoom discussion next Friday as part of this BioEM Talk series. And with that, I thank you very, very much, and I look forward to speaking to you later.